Okay, so let's keep our journey going through cell land. Share my screen, click over here. So this is an active link for section 4.4 in your textbook. And we're going to focus on the endomembrane system and proteins. So basically what we want to know is what are the components of the endomembrane system and what do they do? Okay. But basically the endomembrane system is the packing and shipping industry of our cells. This is the UPS, the FedEx, the Postal Service of our cell city. So this is a series of membranes and organelles found in eukaryotic cells that modify, package, and transport mostly lipids and proteins. And so then our elements are the nuclear envelope, lysosomes in animal cells, vesicles, this thing called endoplasmic reticulum, the Golgi apparatus, and then of course our plasma membrane. So endo means inside. So add that to your little dictionary that you guys are building of what the root words. Okay, so we kind of talked already about the nuclear envelope. We've talked about vesicles. We've talked a lot about the plasma membrane. So we're going to focus this discussion now just on lysosomes, the ER, and then the Golgi apparatus. So the ER is not the emergency room. Okay, endoplasmic reticulum, this is the warehouse. This is where stuff is stored. We might see a few little like finishing touches before it's packed up and shipped off the door. So this is the interconnected sacs and tubes made of a phospholipid bilayer. Yes, we love phospholipid bilayers, they're so cool. And it's actually connected to the plasma membrane. And so it is made of these hollow portions. Like, so the tubes make these hollow portions, these little long tubes of the ER, and then we call those lumen, which means an opening or a passageway. And then it has this other name too, but I think lumen is enough for you guys to need to remember, but you might see it referred to in other sources as this cisternal space. But you can see if we look at the ER here in this image, you can see that it has this highly folded structure, and that's because it's maximizing its surface area to volume ratio, because its job is to move stuff in and out. Okay, so the ER, the endoplasmic reticulum, kind of comes in two flavors. It can come in rough, as the rough ER, or it can be smooth. And really the big difference between the RER, the rough ER, and the smooth ER is whether or not there are ribosomes stuck to it. So in our rough ER, which is what we're seeing here, these little red circles, these little dots, are supposed to be representing ribosomes. So remember that ribosomes are where proteins are synthesized. And so then that means that there's the proteins are being synthesized on the ER, they're moved into the ER, and then they're transported to wherever they need to go through this ER and through the rest of the components of the endomembrane system. And then, so the rough ER is kind of basically producing and modifying proteins, whereas the smooth ER, the parts of the ER without ribosomes, that's main job is to produce and to modify lipids. Okay, and so here's what the rough ER looks like for realsies. So this is electron micrograph. So this is a TEM image, so transmitting electron microscope. So basically you just like throw a whole bunch of electrons on it, and it shows a lot finer detail than what we can see even with our light microscopes. So this is much closer. We wouldn't be able to see this using the microscopes in our lab. But you can see all these little black dots, and that is where proteins are being made. So we see a lot of rough ER in our muscle tissue because obviously we need a lot of protein to compose our muscles. And then they transfer these, the ribosomes on the rough ER transfer the newly created proteins into the endoplasmic reticulum. And then there's some kind of tweaking, some extra folding. Maybe we're going to throw on a side branch on this protein and just make sure that those proteins, you know, there's a little bit of quality control going on and a little bit of refinement before it's sent off to the rest to go wherever it needs to do for that protein to serve its function. And then it can also create phospholipids, which we also need kind of everywhere, as you're starting to see through this endoplasmic membrane system. Okay, so either the proteins are incorporated into the outside of the plasma membrane, or they're transported via vesicles to wherever they need to go in the cell or outside of the cell. Okay, the smooth ER, it's continuous. So we talk about smooth and rough ER, but they are attached to each other. So we might see there's a part of the ER that has a lot of ribosomes, but then the same, if we just keep following it down, we'll see a part where there's not very many ribosomes. We call that smooth ER. So lots of carbohydrates, lipids, and steroids are made in this part of the ER. And it can also act as a detoxification center. So we see a lot of smooth ER in our liver cells and our kidney cells, because that's where the detox is going on. And also we find that there's a lot of calcium ions in these parts of the smooth ER. Okay, and then so one of the other parts of the Golgi, of the endoplasmic membrane system, is the Golgi apparatus. So this is also continuous with the ER. So we can think of this as the postal service. So this is where things are leaving the warehouse and they're going out to be delivered. They're being sorted, they're being tagged, they're being so when things butt off from the ER, they're probably going to make a stop at the Golgi apparatus. And then they're, the book says this looks like pancakes. But so if we look at this micrograph here of the Golgi apparatus, they're like, it looks like pancakes. And I'm like, does it? But I guess if you kind of think of it as them being stacked on top of each other, I don't know. But again, what you're seeing with this, these really highly folded membranes is that high surface area to volume ratio. Again, this is so important. This is why I made you guys do math to understand this, because it is so vital. Okay. All right. And then we have transport vesicles, and those are created either by the ER, usually created by the ER. And then we have this thing called the cis face and the trans face of the ER. And that just means, like, is it close to the nucleus or is it close to the plasma membrane? So the cis face is the face that's facing away toward the interior of the cell, and then the trans face is kind of more toward the plasma membrane. Okay, so vesicles are created by the ER. They go and fuse with the Golgi apparatus. The vesicles fuse with the Golgi apparatus. They dump their stuff into the Golgi, and then whatever it is going, they're sorted, they're tagged, they're modified, and they're giving their directions of where they're going to go be shipped off to, and then the vesicles will pinch off from the Golgi apparatus and either go to and fuse with the plasma membrane, or they'll go off to be wherever these things need to go to. And so if we follow this protein that's made on the interior, the inner membrane of the ER, we can see that if we kind of follow its pathway through all of this, we'll see that eventually it will end up on the exterior of the plasma membrane. Okay, so something that's made on the inside of the ER is going to end up on the outside of the cell. All right. And so you can really just see how these are just transport trucks, right? They're just saying, here's this vesicle that's like delivering it to this stop, and then that center, that shipping center, will go and change it to the next stop. And so this is just like how your Amazon packages get from, you know, the distribution center in, I don't know, let's say Seattle, and then it's transported across the country by going to this distribution center, finally to UPS, and then the UPS trucks deliver it to your house. Same business here. Okay, and then if something doesn't turn out quite just right, it doesn't pass quality control, it might be taken to the lysosomes, which is either the garbage dump or the recycling center, depending on how you want to look at it. Again, lysosomes are not found in plant cells. So we can take that macromolecule, break it down into its component monomers, and then reuse those to build something else. Again, pathogens can be destroyed doing this way. And this is how single cell organisms break down their food, is by tossing it into their lysosomes. Okay, that's where I'm going to end this video. This cytoskeleton will be our last section for this chapter. And so we can practice with these things a little bit more in class on Thursday. So hopefully this is helping, and I'll see you guys on Thursday.